Bismillahirrahmanirrahim. Assalamu alaikum students. Today we will discuss female cone, megasporangium and megaspore formation in ephedra. Female cones in ephedra, these are elliptical structure. Elliptical means it's usually oval shaped structures with pointed terminal part, and it consists of an axis on which the bracts are arranged decussately. Decussately means these are present. The pairs of bracts are arranged opposite to each other, and one pair of bracts is present in one direction, and the other pair of bracts in which the bracts are opposite to each other, this is present at right angle to the first pair of bracts, and the third bract pair is present at right angle to the direction of the second pair. Most of these bracts are sterile, and usually the upper two bracts, these are fertile, and in the axils of these two bracts one ovule is produced on the short stalks. Ovule of ephedra is unique among all the gymnosperms because two integuments are present around the ovule, and these integuments, these surround parenchymatous nucellus, means that inside these integuments the parenchymatous tissue is present, which is known as nucellus. This is the structure of the young female cone, and in this female cone around the axis these bracts are produced in pairs. This is first pair, this is second pair, which is at right angle in direction or position to the first pair of bracts, and the third pair of bract is at right angle to the second pair of bracts. And this is, when it is mature, it is elliptical in shape, and it is pointed at its apex due to the production of the elongation of the micropylar tubes. In this female cone, the lower bracts are sterile, and only the uppermost two bracts, these are fertile, and in the axils of these bracts the ovules are produced. In one bract one ovule is produced. Now we see what is the nature of this ovule in the gymnosperm, which is unique here due to the presence of two integuments. After studying the history of development of the ovule and the internal structure of the vascular tissues, the outer integument which is present around this ovule, it is similar to a pair of the fused appendages or outgrowths, means the pair fused outgrowths are present, which represent the outer integument, and actually it is not an integument, it is the pair of fused outgrowths which is present around the ovule. This is the structure of ovule. Two ovules which are, and each ovule, this is one ovule, this is second ovule, which is produced in the axil of the uppermost bracts, and this is outer integument. which is present on the outer side of the inner integument, and this outer integument, it represents, it is similar like the appendages or the outgrowths which are fused, and it actually is not the integument. These are the two fused uh, um, appendages or outgrowths which are present around the ovule. These ovules are, each ovule is produced on a short stalk or megasporophyll structure, and around this ovule, the ovule which is produced on the short stalk or reduced basal megasporophyll, this ovule is enveloped or surrounded by a pair of fused bracteoles, and the structure of these bracteoles is similar, or these are homologous with the bracteole which are present in the microsporangiate shoot in the male cone. So due to the presence of these characters, the female cone is considered as the compound structure. Now we see what is the structure of ovule. A young ovule, it consists of well-developed central parenchymatous tissue which is known as nucellus, and this nucellus, it is enveloped or surrounded by a fleshy cup-like structure which is usually known as the outer integument, and on the inner side of this integument, outer integument, an inner integument is present which is delicate, and outer integument is attached to the base of the ovule and free above, and inner integument, at the time of pollination, it is prolonged into a tubular structure to form a micropilot tube that projects or it elongates beyond the bracts and the bracteoles which are present around this ovule, and the cells of the inner integument, these contain chloroplasts at the time of pollination. In this diagram, the structure diagram, the structure of the ovule is shown. This is the central parenchymatous tissue which is known as nucellus, and around this nucellus two integuments are present. This is the outer integument which is fleshy and it is cup-like structure which is attached to the ovule at the base only, basal part, and on the above it is free. And then inner to this, the inner integument is present which is delicate, and it is also free on the above part of the above region of the ovule, and this inner integument, it becomes elongated, it projects and it becomes elongated, and it projects above the bracts and bracteoles which surround this ovule, means the bracts and bracteoles which are present around this ovule. And this in inner integument, it elongates more than this and it forms the micropylar tube, and this inner integument, it also remains free from each other and it does not fuse with each other on the above part. At the base of the nucellus basal part, the haustorial region is present which absorbs the nutrition, and this is the region in the center of the nucellus where which is developed after megasporogenesis, when the megaspore, it germinates and produces the female gametophyte in which the archegonia are produced and pollen chamber is also formed. In this ovule, which is also known as megasporangium, megasporogenesis occurs or megaspore formation occurs. How this megaspore is formed in the ovule? In the nucellus, a hypodermal cell becomes differentiated, which is known as the archisporial cell, and it divides periclinally to produce two cells. The outer is known as parietal cell and the lower is or inner is known as the megaspore mother cell. The parietal cell, it produces many cells by repeated periclinal divisions, and due to the production of the new cells, the megaspore mother cells, it is pushed deep within the tissues of the nucellus towards the causal end. Then in megaspore mother cell, reduction division occurs and a linear tetrad of 4 haploid megaspores is produced. Usually the lowest megaspore, it is functional and it enlarges and produces the female gametophyte after germination, and the upper 3 megaspores, these are degenerated. Here's a diagram to show the formation of megaspores. This is the nucellus, and in this nucellus, here the hypodermal cell is differentiated, the diagram of which is not shown here, and this archisporial cell, which is hypodermal in nature, it is divided by periclinal division to produce the parietal cell and the megaspore mother cell. Parietal cell, it again divides many times by periclinal division and it produces the cells. Due to the production of these cells from this parietal cell, the megaspore mother cell, it is pushed towards the lower side, towards the causal end in the deeper tissues. This is the megaspore mother cell, which is enlarged now, and then this megaspore mother cell, it divides and by reduction division, and after reduction division 4 megaspores are produced, 4 haploid megaspores are produced in the linear tetrad, 4 haploid megaspores which are present in the form of linear tetrad, and then from these 4 megaspores, the lowest megaspore, it becomes functional, and then the upper 3 megaspores, these are degenerated, and the lower megaspore, haploid megaspore, which is functional, it enlarges, and then the nucleus of it divides mitotically to produce the female gametophyte, the development of which we will discuss in the next lecture. This is a diagram of the developing female gametophyte. So students, today we have discussed the female cone in the ephedra, which is elliptical in shape, mean oval in shape, and on the terminal part it has pointed end due to the production of the micropylar tubes in the 2 ovules, which are elongated on pollination, and then the bracts which are present in this female cone, these are decussately arranged. This is one pair of bracts, one is in front and the other is on its back side, and this is the second pair of bracts which is present at right angle to the position of the first pair of bracts. Then the third pair is again present at the right angle to the first bract, and the position of this is like the position of the first pair of bracts, and then the fourth pair of bracts is again at right angle to the third pair of bracts, and in this way all these bracts pairs, these are decussately arranged, mean these are opposite to each other, and the direction of the two pairs of bracts, it is at right angle to the pair of bracts which is present just below this pair. And the lower bracts, these are sterile, means no ovules are produced in these pairs of bracts. Only the upper two bracts, these are fertile, and in the axils of which the ovules are produced. In one bract, one ovule is produced. This is the diagram in which the upper two bracts are shown, and in the axils of these bracts, this is ovule of this bract, and this is the ovule of this bract, and these ovules are produced on the short reduced megasporophyll, and each ovule is surrounded by two integuments. This is the nucellus part, and this is inner integument, this is outer integument. These are the structures of the young ovules, and in these ovules, when megasporogenesis occurs, a hypodermal cell, it produces the mm, uh, archisporial cell. Hypodermal cell, which is known as the archisporial cell, it divides to produce the parietal cell and the megaspore mother cell. Parietal cell produces many cells which push the megaspore mother cell towards the lower part of the ovule, towards the clausal end, and megaspore mother cell, it divides by reduction division, and it produces the row of the four haploid megaspores, or a linear tetrad of the megaspores. Out of these four megaspores, the upper three are usually degenerated, and the lowest one, it survives and it becomes the functional megaspore, and these functional megaspore, this will produce the female gametophyte. Uh, Out of these two ovules which are produced in the axils of the two bracts, at maturity only one ovule survives and the other ovule is degenerated, and on maturity, when the seed formation occurs, usually one seed is produced in the cone.